As I mentioned earlier, eukaryotic organisms do not just include algae. It also has protozoans, fungi, plants, and animal cells. The cells of the eukaryotes are structurally much more complex and much larger in size as compared to a prokaryotic cell. There are some major differences between the two types of cells, which makes it more apparent, and let's look at those differences up here. If you recall, the number of cells in a prokaryote was just 1, and eukaryotic organisms can have multiple cells. That means it can start from 1 to many cells. The DNA shape in prokaryotes is a circular DNA with no histones. Histones are the proteins that anchor the DNA. These are present in eukaryotic cells. So eukaryotic cells have the if this is the helical structure, then each of these is anchored around a protein that helps it make more compact. Think of it as a spool and a thread that's going around the spool. That's how the consistency of the histones are, and that's how it gives its structures. Those are present only in eukaryotes, and they're absent in prokaryotes. Prokaryotes, DNA is free in the cell. Eukaryotic DNA is bound in the nucleus. There are no true organelles in prokaryotes. Yes, there are organelles in eukaryotes. Very important feature are the ribosomes. If you remember, I said earlier also that antibiotic can affect bacterial activity because of destroying the ribosomes. The bacterial ribosomes, if you recall, had a 50S and a 30S, two subunits. Eukaryotic ribosomes have a 60 and a 40S subunit. So S again stands for sediment. It has nothing to do with addition of the two subunits. This is the one unit is 40S and the other one is 60S, and these we have 30 and 50S subunits. The first amino acid in proteins was synthesized in the eukaryotes is the methionine. While in prokaryotes, it's the formyl methionine, which is the first amino acid that was formed. Cell wall of a eukaryote will depend on what particular cell it is. For example, if it's an animal cell, it will have no cell walls. If it's a plant cell, then it's going to be composed of cellulose, and if it's a fungi, then it's composed of a material that is called as chitin. On the other hand, the cell walls of prokaryotes have peptoglycan, and peptoglycan is sometimes referred as PPG. Reproduction for prokaryotes, they reproduce by an asexual means of reproduction, and that is binary fission. In binary fission, a cell can divide either horizontally or vertically, and it can give rise to, two cells that are exactly the same. Eukaryotic can be either mitosis or meiosis. And meiosis is the kind of cell division that some of you probably have already studied these two in the previous biology classes. So those are the basic differences between the two, and it's really important that you do understand these differences and you're able to give me the details on a test or so forth. Let's look at the next slide now. Do they have flagella? Flagellas are structures that are present outside the cell wall, and the flagella pretty much helps in motility for these cells. The pictures up here on the left is a flagella that's present in the euglena, and the other one is actually a cilia that is shown in the I think it's a tetrahemina. That's what it is. That's where it's shown as to what this one cell, this particular one, is. These are actually, sometimes they also have cilia. So this is actually they're showing lots of different hair-like structures, and these are all cellulose. And this is the flagella. Both are made up of microtubules. Microtubules are long, hollow tubes, and these tubes are made up of a ring in the center. For example, over here, notice there are 2 rings in the center, and they're surrounded by 9 rings. So that's why it's called as an axial pattern of 9 by 2. The flagella in the eukaryotes can rotate. Unlike the one in prokaryotes, well, actually, the prokaryote flagella rotates. The eukaryotic flagella has a more wave-like pattern to it. So these flagellas can be used for whipping and pushing and forward and so forth. Do they have cell walls? As I already mentioned before, plants have cellulose, which is a composition of a carbohydrate. Fungi have chitin, and protists have something called as pellicle. So they all have different cell wall. They do not have PPG, which is a peptoglycan. Their cell walls are chemically much simpler than the prokaryotic cell walls, and that's the reason that antibiotics do not harm eukaryotes, because antibiotics are designed to destroy and break down the configuration or the lattice structure in the peptoglycan. Some of these they also have a glycocalyx, some of these cells, and glycocalyx is a layer of material that is a very sticky carbohydrate, and it pretty much helps it's like a protective coating, because the eukaryotic cells, for example, animal cells, only have plasma membrane. So the glycocalyx will provide strength to the eukaryotic cell. Okay, let's now look at the next structure, and which is the plasma membrane. Plasma membrane is very similar to prokaryotes. That is, it's also phospholipid bilayer, and it is embedded with, however, it's embedded with cholesterol and proteins. And it also follows the fluid mosaic theory. Now, you recall the fluid mosaic theory is pretty much is the concept where we are saying that the pattern of proteins and cholesterols are impregnated within the plasma, the phospholipid bilayer. So the phospholipid bilayer is just not by itself. For example, if these were the phospholipid bilayers, then in between the phospholipid bilayers, you will have segments that are either composed of cholesterol or proteins. And they give you on the top, if you were to then take cross sections at this section, then a cross section will give the appearance of a fluid, of a mosaic. So that's why it's called as a fluid mosaic theory. Now, fluid is because it is also dynamic. It's a very fluid-like structure, just like if you were to take a little bit of dishwashing soap and you put it in water and you see it floating around. That's the consistency of a plasma membrane. The plasma membrane is also semi-permeable, which means that it will allow certain molecules to pass through them, and it is selective, which means that it allows certain molecules to go in and certain to go out. So it's selective to which part will it allow to go in and which part to go out. Substances can come in and out of the cell through the process of endocytosis. Endocytosis is pretty much, it's a mechanism by which the cell can engulf food particles or solids and so forth. So, for example, up here you can see how the membrane is protruding out to eventually enclose the substance and it's taken in. Now, endocytosis is, endo means inside, cytosis is eating. Exo is outside. So substances sometimes have to be also removed from the cell, and that follows the pattern of exocytosis, where the substance can actually, for example, if there was a molecule here, it can actually travel and come close to the plasma membrane. Plasma membrane can then engulf down, and then eventually this can be thrown out of the cell. So that would be exocytosis. What's different about the cytoplasm? The cytoplasm of prokaryotic cell is composed of three components. A cytosol. The cytosol is the fluid that is present in there. Organelles are all the structures. So, for example, the nucleus, the endoplasmic reticulum, the chloroplast, the mitochondria. These are organelles. So these will be all present inside or suspended into the fluid. And the cytoskeleton refers to substances that are present in the plasma membrane, and these cytoskeleton are actually microfilaments, microtubules, and they give an internal structure to the cytoplasm. And there's another kind also, and that's called as the intermediate filament. These are all long protein tubes, and they help the cytoplasm to move from one part of the cell to the other. Imagine this is more like a churning machine, that it can help distribute substances from one side to the other, and that movement is called as cytoplasmic streaming. What about the organelles? I mentioned earlier that these are structures that are present or suspended inside the plasma membrane. So these each organelle is to be a true organelle, it has to have a membrane bound. And eukaryotes have organelles, and I'm going to highlight some of the organelles over here. Let's go with the mitochondrion, chloroplast. Some people consider these reticulums as also organelles, and then the nucleus. These are the basic organelles that we can see up here. And I'm trying to see lysozyme, that's also an organelle. So these are all different organelles. Let's look at the first one, which is the nucleus. The nucleus is the most characteristic organelle that you see within a cell. The nucleus is usually spherical, or it could be oval, and it is the largest structure in the cell, and it contains almost all of the cell's hereditary information. So it is the director of DNA synthesis. It allows, just before a cell has to divide, it directs the synthesis of its DNA. The nucleus is composed of a double membrane, which is called as the nuclear envelope. The nuclear envelope resembles the plasma membrane in structure, except that it has tiny channels in the membrane, and these are called as the nuclear pores. The nuclear pores are openings through which substances can move from the inside of the nucleus to the outside. So it pretty much helps in movement of certain molecules and certain substances, and also controlling what can go in and what can go out. So it's sort of communication gateway for the substances. That's the nuclear pores. The next important feature within the nucleus is called as the nucleoli. The nucleoli are actually condensed regions that's where a special kind of RNA called as the ribosomal RNA is synthesized, and the ribosomal RNA is actually essential for structures that are called as the ribosomes. So, in other words, the nucleoli helps in the manufacturing of ribosomes. And lastly, we have a thread-like mass, which is called as chromatin. Chromatin is the thread-like mass that is present extensively, that is shown up here. All these little dark or purple regions that you see are all chromatin fibers, and the chromatin fibers are composed of DNA strands, and these actually taken just before a cell divides. So I'm going to show you on the next slide how that takes place. So let's look at the DNA up here. You can see the DNA is shown up here as a double helix. The DNA in a cell could be 2 meters long. So the DNA is then wrapped around these proteins, and which I was talking earlier, imagining a thread being winded around a spool. Those are called as the histones. So these are proteins. These proteins are actually negatively charged, and DNA is positively charged. So it gives you, it helps to be compact. Now these compact regions are called as condensed nucleosomes, and they eventually form what is called as chromatin. Chromatin is a fibrous form, a hair-like, thread-like mass, whatever you want to call it. And chromatin then eventually condenses more to form what we call as chromosomes. So these are the 3 different terms that you should understand. The DNA is a double helix. It is present as a thread-like mass within chromatin, and just before cell division, which could be either mitosis or meiosis, the chromatin then forms the shape of a chromosome. Now, both of these processes of mitosis and meiosis are not present in prokaryotes. Therefore, they don't have this structure of the DNA. Another organelle that we want to look at is the endoplasmic reticulum. Endoplasmic reticulum within the cytoplasm of the eukaryotic cell is actually an extensive network of flattened membranous sacs or tubules, and each of these tubules are called as cisterns. And the ER is actually continuous from the nuclear membrane. If you notice up here, you can see the outer nuclear membrane actually invaginates outside to give rise to this extensive work. The endoplasmic reticulum are of 2 kinds. There is one kind that is that has little dots that are shown over here, which are the ribosomes. So they're embedded with the ribosomes. Ribosomes are protein manufacturing regions, and ERs that have ribosomes are called as the rough ERs, and the ERs that do not have ribosomes are called as the smooth ER. Now, the rough ER is where proteins will be synthesized. That's where, and the smooth ER is helpful for lipid manufacturing. Usually phospholipids and steroids and estrogens and testosterone are all synthesized within the smooth ER. In some cells, for example, the liver cell, sometimes the smooth ER also helps in the formation of glucose or some carbohydrates. What are ribosomes? We just learned the name previously. Ribosomes are structures that some call them as structures, some consider them as organelles. So there's a controversy on them because they really don't have a double layer membrane. That's why I would just say it's a structure. But if somebody disagrees with that, that's fine. Some books will call it as organelle. The ribosomes in eukaryotes are made up of 2 units, a 40S and a 60S. So there's a smaller subunit and a larger subunit. Together, the sediment at 80S. Remember again, there's no math here. It's the sediment weight of these 2 subunits. What is it in prokaryotes? Does anybody remember? The answer is 70S. The chloroplast and mitochondria they have ribosomes which are more similar to 70S, and that's why we will talk about the endosymbiotic theory later, but just wanted to bring that to your attention. Okay, the next organelle that we want to look at is the Golgi Complex. Golgi Complex are actually, these are transport vesicles, you could say. Most proteins that are synthesized by these dots, which were the ribosomes, are carried through endoplasmic reticulum, and they end into these vesicles that are called as the Golgi apparatus. So here is the entrance to the Golgi apparatus, and here is an exit. And by the time the molecule is exited, notice it's been packaged. So the transport vesicle is eventually a surface that helps transfer the proteins. It modifies the proteins. It packages them, and if these proteins can either be transported to be taken outside the cell, or they can be used as storage vesicles in certain cells, in certain cases, because these proteins can be used later. So this is the packaging and the transport region. Lysosomes, another organelle. Lysosomes are sort of, they originate originally from the Golgi Complex, but they're enclosed spheres, and they, unlike mitochondria, they only have a single membrane and they lack internal structures. But there are 40 different kind of lysosomes. So there are different kinds of lysosomes. They have many different kinds of enzymes, and these enzymes can ingest bacteria as they enter the cell. Cells that are more active and they do undergo more phagocytosis. For example, the white blood cells will have many more lysosomes in them. What are vacuoles? Vacuoles are space or cavity in the cytoplasm that is enclosed by a membrane, which is called as the tonoplast. Tonoplast is the membrane that encloses this membrane over here. And they actually will have somewhere from 5 to 90% fluid in them, depending on which cell it is. If it's a plant cell, it almost will be 90% of the cell would be vacuole. In a plant cell, if I was to draw a plant cell, the vacuole would be this large. In other cells, you can see the vacuole being smaller. So the vacuoles may derive from Golgi apparatus again. Some can exist as storage, some exist as water retention, some have molecules such as proteins, sugars, and amino acids, and so forth. Other vacuoles can be formed during endocytosis to help bring food into the cell, and many times poisons can also be stored in these vacuoles. So all kinds of different vacuoles. The next feature we want to look at is a mitochondria, which looks like a little football. Actually, I don't know if you want to call it as a football or a kidney. It's more like a kidney shaped or a rod-shaped organelle, and it's present throughout the cytoplasm of most eukaryotic cells. Certain cells have more mitochondria depending on their need, because if cells have to generate more ATP, they will have more mitochondria. The mitochondria has two membranes. The outer membrane is smooth, as you can see in the picture over here. This is the outer membrane. The inner membrane is the outer membrane is like this, and the inner membrane has folds, and these folds are called as cisternae, and cristae. These crista is the singular. Cristae are the fold that will make up. Now, the fluid part, which would be inside over here, the space is called the matrix of the mitochondria, and it is actually made up of it's a semi-fluid in nature. The inner membrane having folds gives a very large surface area to some certain reactions which generate ATP, and we'll talk more about these reactions later in the coming chapter. Mitochondria are also called as the powerhouse of the cell. That's where the energy or ATP is being generated. A lot more about this later. What are some other organelles? Another organelle that we want to look at is the chloroplast. Chloroplasts are present in either in algae and in green plants, and it's a unique organelle. A membrane. It's a membrane-bound structure. This one actually looks more like a football. It has lots of greens, and that's the chlorophyll. It has stacked of these regions that are called as thylakoids. Thylakoids are more like pennies. If you have a lot of pennies that you're stacked up on top of each other, that's how the thylakoids are. And these stacks are all together called as the grana. So you have thylakoids, each thylakoid stacking up as grana, and then granums, many granums together make. Grana is the singular, granum is the plural, and they make the structure within the mitochondria, right? Within the chloroplast. Sorry, the chloroplast also has 70 S ribosomes. It has its DNA. It has enzymes, and this gives you another reason to understand that they were once an autonomous structure by themselves, and with time they were taken in through an endosymbiotic theory, and we'll talk about this in a couple of next slides. Lastly, what is the cytoskeleton? Cytoskeletons are composed of microfilament or microtubules, and both of them, I told you earlier, were tubes of proteins, and they provide pathways, they provide structure to the cell. So here is your microfilament. You can see the microfilaments are broader, and microtubules are more like railroad tracks. They also help movement of particles from, for example, this part to go here. It can walk through or pass through these microtubules. The endosymbiotic theory is a theory which explains how once upon a time in a very early stage of Earth, the prokaryotic cell, for example, chloroplast and mitochondria, were once upon a time a prokaryotic cell or a bacteria by itself. And according to this concept, this was engulfed by a larger bacterial cell, and eventually there was a symbiosis between the two cells, and this symbiosis means living together. So when they live together for a long time, then the chloroplast and the mitochondria actually became a part or integral part of a eukaryotic cell. And that's why we see that both of them have their own DNA. They have 70 S ribosomes, and they can divide within a cell by itself. So these are some unique properties of these two organelles, chloroplast and mitochondria. So we'll stop here. This would be your first lecture on eukaryotes. There's another one to follow this one.